very basically with plant sciences. So we as plant scientists, we are interested, of course, in securing the availability of food, because all our food comes from plants, whether we eat the plants ourselves or whether we feed them to our livestock. And the secure supply of food is massively endangered by rapid climate change and even more rapid population growth. What classical breeding technologies do and did is try to make our crops more resilient to all of these stresses that come with these changes in our climate. For example, in this image I tried to generate with ChatGPT here, you see a tomato cultivar that has nice and big fruit, but it seems to be susceptible to a pathogen, for example. And what breeders very often do is go into closely related land races, so species without like these economic value, and try to look for, in this case, for example, for resistances to this certain pathogen, and then breed these resistances into these elite cultivars to get bred engineered cultivars that have all the desirable traits. And this is what's being done by classical breeding. But as molecular scientists, we of course think that we can speed this up and make this easier and more accessible with genome engineering and genome editing techniques, to be also able to use natural variation and interesting traits, could be resistances, could be other traits from also more distantly related plants, and engineer them into our interesting crop plants. All of this and all of the genomic basis that we need, and the genomic understanding that we need to do this, we have from Arabidopsis thaliana. So Arabidopsis thaliana is, for us plant scientists, is the model organism. And I called it like the Rosetta Stone for plant genomes, because it told us basically, let's say, all we know about plant genomes to date. Arabidopsis has a small diploid genome, has very low repeat content, is very accessible in the lab, so it's easy to transform, has a short generation time, so we can really nicely work with it in the lab. And for decades now, it taught us a lot about key pathways in plants, such as flowering time, hormone signaling pathways, the development and response also to environmental cues of a plant, for example, in response to biotic and abiotic stresses. So we learned all of this from Arabidopsis, the model plant. But if you compare Arabidopsis thaliana, our model, to actual crops, you see that crop plants are much more complex, especially on the genomic side. And we need to understand these genomes to do these nice fancy modern genome editing techniques. So crop plants have often much larger genomes, have a higher content of repeat elements, and also in the lab they are not always so easy to access like we can do with Arabidopsis. Like they are easier, like they are much harder to transform. They have longer life cycles, and you can actually really see that their genomes, were shaped via this artificial selection, via breeding over centuries now, rather than this natural selection. And especially this selection has been done for yield traits now over centuries. Many of these traits that we see in crop plants are also very often polygenic, so very complicated. They are not regulated by single major alleles, but very often by a stack of small quantitative alleles that together add up to make these traits. So in order to optimize crops, like for example by genome editing, we have to find these traits, and then it's very important for us to link these traits that we see in the phenotype to the genomic variation. So to understand the genome, to understand the traits, to be able to engineer them. And to understand genomes, to understand genomic variation, we of course need this genomic variation. We need many genomes to try to understand them. So people go out into the wild and collect plants and screen them for interesting traits, and then we can sequence their genomes to try to understand these traits. And one very interesting thing is that it's, I think, especially for you as sequencing specialists, you know that sequencing sometimes is the trivial part. So it's very easy to sequence plants, but especially for plants, it's sometimes much harder to actually get to the DNA. So some plants really do fight to release their DNA, and many people work hard to, in the first place, get to the DNA and then the sequencing and also the analysis might be the more trivial part. And to make this, maybe a little bit more trivial, I'll today also show you some tools how we can make our lives easier in terms of analyzing genomic variation. I want to show you one example from my PhD where we analyzed genomes for interesting variation. No, I will do this later. Before, I will show you that we have now this genomic variation. So we have now, 2026, we have over 2,500 genomes, not necessarily all from individual species, but from many, many, many species and subspecies. We have genomic variation, and is constantly rising with the ease of available sequencing techniques. Now back to the example that I promised to show you. So in my PhD, we sequenced a variety of genomes from a family called Brassicaceae. So Brassicaceae are crucifers, most often little weedy plants. And you may see here our model plant, Arabidopsis thaliana, in there, but you may also see species like Brassica oleracea, that's cabbage, and here down there, lesser known plant, Diplotaxis tenuifolia. You know this as arugula or rocket. So some of these plants, for example, arugula, have interesting photosynthesis traits that we think might be more efficient, especially under heat and drought. And in my PhD, we sequenced 19 of these species with this variation in the photosynthetic traits. And now the question was, okay, what do we do with these genomes in the first place? Because in the first place, we have to understand what these genomes consist of, like we have to annotate them. And usually for this, you need RNA-seq data. For example, it could be short-read RNA-seq data, could be these more advanced iso-seq data, long-read data that we now have to annotate these genomes. But the problem is that you need RNA from different conditions to like capture the whole genic space of these genomes. And people from my lab, together with the Usadel lab at HHU, and I was also fortunate to contribute here a little, developed a gene structural annotation deep learning tool that is called HELIXA. And HELIXA is trained on known annotations for genomes. And while for these normal wet lab procedures, annotating genomes, as I said, you need RNA-seq data, HELIXA only needs a genomic sequence file, like a FASTA file, that you put in, and you get your annotated genome as a GFF file. And here in this image, you see the architecture of HELIXA. It's a complex deep learning architecture. We'll later, for another example, try to understand some of the algorithms that are being used there, and try to introduce some of the algorithms that are being used also here in HELIXA. So you basically put in your genome and you get a nice high quality annotation of these genomes. So we did this for our genomes. The genomes from the plants with the interesting photosynthetic variation. And we see here a nice variation in genome sizes. And we then also used another algorithm that is called EDTA to annotate transposons. What you see here is that high like sequence space in these genomes is covered by transposable elements, so by repeat elements. We could nicely annotate here in orange, we could annotate the genes. And then you see that there's also not annotated space in these genomes, so sequence parts without annotation. Two things in this graph that I want to put your attention to. Like, look at Arabidopsis thaliana, look at how massive this outlier is. I mean, this is our model plant, and you see it's completely different from many, many other plants that we know. It has a much smaller genome and a highly reduced content of, for example, transposable element. It's very interesting to see that so much that we know about plants relies on Arabidopsis, where Arabidopsis looks so much different than plants that we might be interested in. Second example that I want to show you is this gray space that you see everywhere here in these bars. And this is the unexplained sequences or the unannotated sequences that these genomes contain. People used to call this like junk DNA or like genomic dark matter or whatever, but actually this is the regulatory DNA, so the DNA that regulates all the genes. And very often this contains very promising targets. So this regulatory DNA contains very promising targets. And it's known that regulatory DNA was very frequently the target of breeding, of natural selection and breeding selection. And we think that we can also use this regulatory DNA to engineer genomes towards interesting traits. And I'm gonna, in the end of this talk, I'm gonna show you some examples how we think we can target these regulatory DNA. But why is regulatory DNA so important? And this has all to do with gene expression. And for the rest of the talk, I'm going to talk about gene expression here. So why are genes expressed differently? Why is gene expression, like transcription, why is this so tightly regulated to make a tightly regulated quantitative control of gene expression? I want to introduce what we call the grammar of gene expression, and this is not necessarily only for plants, but this is a general model of how gene expression works. So imagine two genes, here gene 1 and gene 2. In our graph before, they would have been the orange parts of these graphs, so that's the gene space. And these genes are here now quantitatively differently expressed. So gene 2 has a higher expression. Why? This is because of the gene regulatory elements that are in between these genes. So the easiest type of gene regulatory element is, of course, the core promoter. And the core promoter is basically just the part that sets the area for the transcription start site and confers a very basic level of transcription to this gene. And these core promoters, or the strength of these core promoters or of expression in general, can be enhanced by these enhancer elements. These are genomic elements that increase the level of transcription of single genes, for example, by binding of transcription factor binding sites. And in the other way around, there are also silencers, and these silencers reduce the level of transcription. You see in this case, gene 1 is now off because there's a silencer in front of the gene. And yeah, these silencers reduce or even completely shut off genes, also by binding of transcription factors. Then there's also something that is called insulators in plants. And actually in plants, we don't really know how insulators work. We don't know whether things are binding there or whether this is DNA structure stuff here. But these insulators should block the activity of, for example, enhancers to genes they shouldn't be regulating. So in this case, this enhancer should only regulate gene 1, but not gene 2. This is why in plant genomes there are these enhancer blocking insulators. And all these elements, they work together to make this tight control of gene expression. Most elements here even contain multiple functional sites. For example, these enhancers here, and in this talk I will focus on enhancer sequences. These enhancers here can contain multiple transcription factor binding sites. To make it even more complicated, not all of them have to be bound at the same time. So it depends on whether a transcription factor is present in the cell or not, and also on the affinity that transcription factor binds to these functional sites in the enhancers. So the whole pattern here is very, very complex. And this complexity leads to this very strict regulation that is time specific, that is condition specific, that is tissue specific, and that is also cell specific, right? So this is how these elements together make this very, very tightly regulated specific control of gene expression. And this is very poorly understood in plants. So this is like the hypothetical model. We know that these elements exist in plants, but for many cases, we don't really know which elements are working together to make this expression and how this goes to, or how this mediates or establishes interesting traits. So we need a method to quantitatively unravel this regulatory activity of the different elements. Yeah. Best case in a very parallel high throughput fashion. And this is why the lab that I'm right now doing my postdoc in developed this technique that is called Plant STARR-seq. It's a massively parallel reporter assay where you, in a high throughput fashion, can analyze all these different types here of regulatory elements. And for the explanation here, I will focus on the enhancer elements. Yeah. So let's say we have a library of enhancer elements that we want to test for strength. Yeah, we want to see how strong these enhancers are in a plant. So we clone these enhancers here upstream of a core promoter, upstream of a GFP gene. We do not want to use this GFP gene for fluorescence. We are not interested in the fluorescence. We just want to have something expressed under the control of this enhancer. We just want to analyze the RNA of this GFP gene. And this is because we barcoded this GFP gene. So this little bar here is the barcode. And before we do the experiment or in preparing the library, we link this barcode to one specific enhancer. So we know which barcode corresponds to which enhancer. And then we take this big library, and to talk a little bit about numbers here, we did a screen for enhancers where we screened around 350,000 enhancers in one go, in one experiment. And we take this big library and we can either transform tobacco leaves with this. So in plants, you can do this transient transformation where you basically inject bacteria into the plant leaf, and then the genes are expressed in the plant leaf. So we can either screen this library in tobacco leaves or also in maize protoplasts. And then from these systems, we can extract the mRNA and sequence the barcodes. And the abundance of the barcodes should then correspond to the strength of the enhancer. For example, let's take this blue enhancer here, this blue enhancer. We tied this to this purple barcode. Then we can count the barcodes here. Let's say we have 4 times more barcode here than we had in the input DNA. So we have a log2 enrichment, and this is compared to a no enhancer control. So we clone in one core promoter without an enhancer. This is our control, and we compare the strength of our enhancers to this no enhancer control. So in this case, we have 4 times the abundance of this interesting barcode here compared to the no barcode control. So a log2 enrichment of 2, which is our quantitative readout of the enhancer strength of, for example, this blue enhancer here. And to show you a little bit that this assay works and the dynamic range, we just took here 4 enhancers, 4 different enhancers from different species that we tested in this enhancer STARR-seq, and you see it has a very reproducible and very high dynamic range that we get as an output here of the log2 enhancer strength. So as I said, our enhancer screen contained many enhancers. So 350,000 enhancers we screened in one go, and we screened them under various conditions. So the first 4 conditions here, light, dark, warm, cold, were the conditions of the tobacco plant after the infiltration. So we infiltrated this library into tobacco, and then we kept the tobacco plant under these conditions. And the last 3 conditions were the library transformed to the maize protoplasts. So you have many different conditions and many different sequences. So we have a huge array of data points. So over 4.5 million data points of this enhancer screen. And when you have an easy input, which is our sequence, and an easy output, this always screens for deep learning. So when you do the experiment, what you get is you get your 170 base pair sequence here. It's like we define input for Plant STARR-seq as 170 base pairs because that's like the technical limitation of our assay. It has something to do with how we prepare these sequences. It's always 170 base pairs for us. And when we do the Plant STARR-seq experiment, we get the strength of the measurements under different conditions. But since we have so much data, we can do machine learning techniques here. Yeah. And now I want to give you a little introduction into deep learning, but please keep in mind, I'm a biologist that somehow ended up in front of a computer, and I tried to teach this to myself, and I'm now trying to give you a little introduction into deep learning, but please take everything with a grain of salt here, as I'm a biologist. So the first thing that I want to show you is how we can actually feed a computer these sequences in a machine readable format. Yeah, because the computer is very bad at reading letters. It loves to read ones and zeros. So the first thing that we do in machine learning is that we convert our DNA into letters of ones and zeros. And this is by a technique we call one-hot encoding. So we have 4 DNA letters, ACGT, and we simply give each of them one vector out of 4 bits. And of these 4 bits, only one can be 1. This is why it's called one-hot encoding, because just one bit in this vector is hot as 1. And by doing this, we convert our sequence stretch. Here I gave you a sequence stretch of a few sequences. We can convert this into these vectors, and we can combine them into a big matrix that is now only ones and zeros. And the computer likes to read ones and zeros. And then we can feed this matrix here, this one-hot encoded matrix that contains our sequence, into a very, very basic and easy type of machine learning algorithm. And this is the most easy type of machine learning algorithm, that is called a perceptron. Very easy, deep neural network, a perceptron. And actually, perceptron is so easy that you can actually run it in Excel. It's pretty easy. This perceptron contains of multiple layers. The first layer is the input layer, and the circles that you see here are called the nodes. And each node just holds one number. So we can put our matrix, our sequence matrix, into the input. Yeah. See here, the first vector is the A, and the second vector is the G, and so on. I was very space limited, so I just did it for 2 bases here. And then what happens in this perceptron is that this vector here gets multiplied with a matrix, and this matrix, it's called the weights. And the multiplication with this matrix leads to a second layer, it's called the hidden layer, and by further multiplications, in the end, you come to one output layer, which is just one number. And all in between is just matrix multiplications. These matrices here, these weights, initially they are completely random, and so will be your output. This output could here, in our case for the STARR-seq, for example, could be like the strength of your enhancer under one condition. Before with Helixa, with our gene structural annotation tool, it could be like the probability that this sequence is an exon or an untranslated region. This is like the output. And I said initially, this output is entirely random because the weights are random. And in a process that is called training, these matrices are adjusted to somehow make this value here match your actual output on your training data. So you know your output from your training data, and you make these weights, you adjust these weights here to make your output match your training data. And then in the end, all your weights is your model. That's the simplest form of a deep learning neural network. But of course, this is super simplistic, and this model will not lead to anything because it doesn't now know like the core features that are in DNA, and these are very often motifs. So DNA is full of motifs. Could be motifs that define an exon-intron border, could be motifs that define an untranslated region, or could be motifs, and we are interested in these motifs that are, for example, transcription factor binding sites, because they are interesting in plant gene regulation. And to find these motifs, what we use is a so-called convolution. So very often we use these convolutional neural networks. And these convolutional neural networks, they come actually from computer vision. Imagine this input image, this like a representation of, let's say, the letter O as pixels. Yeah, so it's a ring of pixels here representing the letter O, and the computer should now learn that this is an O. What convolution basically is that you take a so-called filter, some people also call it a kernel, and you slide this over your input image. And this filter contains a little pattern. For example, this pattern here matches the first quadrant of your letter O. And then the output of this convolution is a so-called feature map, where you see, okay, in the first quadrant here, we have a perfect match of this filter to our input image. And also in the lower right quadrant here of our input image, you have a perfect match of this filter with the input image. This is why in the feature map, basically in the result of this convolution, we get a high number. So this feature map is now a very abstract representation of our input image and our filter. But this now actually contains information about motifs that we see in our data. And instead of putting our input image into a perceptron, we can now put our feature map into this perceptron. And this, the output of this perceptron might now be, yeah, I'm 80% sure that this is an O. Might also be, yeah, I'm 20% sure that this is an elephant. But yeah, depends on what you train your data with. Of course, when we do genomic deep learning, we do not do this with representations of bits on a screen, but we do this with our one-hot encoded sequences, with our matrices here. So again, a little sequence stretch, one-hot encoded. And the same as we did it with the letter O before, we can now apply a kernel, a filter, that slides over this matrix, this one-hot encoded matrix, and gives us this very abstract feature map that contains information about whether we see the pattern in the filter in our data or not. And then again, instead of putting our one-hot encoded matrix into the perceptron, we can put the feature map into the perceptron and then get as an output, for example, predictions of how strong this enhancer is that we see here. And this is, we do not do this, of course, for one single sequence and one single kernel and one single feature map, but we do this massively parallel. So we do this for hundreds of thousands of sequences and for millions of kernels and get millions of feature maps that we feed our deep learning algorithms with. And when we do this for STARR-seq, I just told you, instead of doing the Plant STARR-seq experiment that gets you the measured data, we can do the one-hot encoding of this data, put this into our convolutional neural network, and get a prediction. And again, in the first place, because everything is random here, also these filters here, in the beginning, they are random. We have the algorithm learn themselves. They are random. And then we compare them to our measurement, adjust the weights until we get a better representation. So we get a better prediction of how strong these enhancers are compared to the measurement. And this here is called training. So we take our whole library, the data from our whole library, and train these convolutional neural networks with them. And the aim is that in the end, when you have an untested sequence that has never been in a Plant STARR-seq experiment that you want to analyze, I could send now this trained model to you. You one-hot encode your sequence and get your enhancer strength prediction of your genomic data. This is like the way we intend to use this convolutional neural networks for Plant STARR-seq data. And we come to accessibility of these models later, because actually this is still very computational. You have to have these models, you have to have the software to do this, but I'll later show you how this can be made much more accessible and what we're doing on this side right now. To convince you that these predictions actually work, we only took 90% of our big 350,000 enhancer screen for training the model. So we kept 10% to ourselves that the model has never seen, and then predicted the strength of these 10% of the data and compared it with the measured data from our screen. And you see a very nice and high correlation. So we can actually explain the measured enhancer strength to around 70% with our prediction tools. So we have a correlation of around 70%. And I actually think that 70% is basically all you can explain by sequence only in terms of enhancer strength. I think the rest is stuff that is not in the primary sequence. So DNA modifications and epigenetic stuff and so on. I think this is basically the boundary of what you can explain with deep learning only. I always showed you, when I go back for a sec, these convolutional neural networks here as a black box, because very often people say, yeah, that you cannot know what these models actually learn. So you cannot know what these weights and kernels actually do represent on a biological level. But that's not really true. We can very well open these black boxes and try to understand what the model learned here. And there are special tools for this. For example, this tool called DeepLift that lets you screen sequences for like individual bases that the model thinks are relevant for, in this case, the strength of the enhancer under a certain condition. And this is basically what happens if you put one sequence into this tool. You get for each base of the sequence, you get a contribution score of the enhancer strength under a certain condition. And very often these patches here of highly contributing sequences, they are very often transcription factor binding sites. So the model learns to find the actual biology, so relevant functional sites by itself. Yeah. And I think an advantage here is to use these tools, these predictions and these algorithms here as a tool to annotate functional sites in whole genomes. So for example, transcription factor binding sites. There are tools that do this already, that scan your sequence for transcription factor binding motifs, but they never do it in a condition-specific way as we can do with these Plant STARR-seq models. Yeah. So for example, here you see that these two interesting functional sites, they only pop up in the dark condition. So the model learned that these sequences here only have a high attribution to the enhancer strength in the dark condition. And this is something that I think you cannot easily do with these straightforward genomic transcription factor binding site annotation tools right now. I want to give you a short summary after this deep learning part, so everyone who fell asleep during this math bit can wake up again, because afterwards we'll shortly do some more biology. So Plant STARR-seq is a very nice technique to accurately determine enhancer strength, not only strength of enhancers, but actually we can also do this, for example, for silencers or other interesting regulatory elements. We can use the data from Plant STARR-seq to train deep learning models and use them to predict the enhancer strength without having to do the actual experiment. And by opening this black box, these deep learning models, we can identify underlying sequence motifs in a condition-specific manner. And now, of course, how does this apply to actual use cases? And I brought two case studies here, one from my PhD and one other case studies where we think this actually translates into interesting genomic variation. I have to drink a bit of water before showing you this. So, first case study is from my PhD. These here are genomic elements from two of the species I showed you before in the species tree. Like, remember, we had this 19 Brassicacea species that we screen for interesting photosynthetic variation. And two of them we have here. So we took the upstream regulatory region from two closely related species that have very different photosynthetic types. The lower one we think is more efficient. It's so-called C3, C4 intermediate photosynthesis, in case you want to go into this in depth. And we think that species with this photosynthesis trait can be more efficient, especially under heat and drought conditions. And the gene here that we were interested, also a little side note, is called the GLDP1 gene. And we know that this is a major allele which spatial variation in expression contributes to this trait. And we see that the regulatory region of this gene, in the one case, leads to expression of, in this case, a GUS reporter gene in the whole leaf. But in the species of the same region from the species with this interesting variation in the photosynthetic trait leads to expression of the reporter gene only around the veins. And on a biochemical level, this makes a lot of sense. But of course, we were interested in what is going on here in the upstream regions that leads to this very distinct spatially differential gene expression. And over many publications, and also now a little bit with the help of these deep learning tools, we now have a model where we could pin down two potential enhancer sites, both with different transcription factor binding sites that are present in both upstream regions, where in the upstream region of the plant with the interesting variation, we have an insulator here, a transposable element. that seems to have enhancer blocking activity as an insulator of one of the elements. And this current hypothesis may lead to this interesting variation of spatial gene expression. So one of the use cases where we can use this knowledge to maybe potentially engineer plants towards interesting photosynthetic traits. Another use case has to do with tomatoes. So there is this trait called locule number in tomatoes, and locules are these pockets here in the tomato that contain the seeds. And apparently it is of breeding value to have many of these locules, so to make nice and big tomatoes. In some markets, they seem to go really well. So for certain variety of tomato, we want to have this big, large, many locules. And in 2021, people from Cold Spring Harbor published a study where they could pin down variation in this locule number to one major allele, actually, again, to one gene regulatory sequence in the upstream region of a gene. So this gene is clavata3. It's one of the textbook developmental genes in plants. And people have shown that if they delete part of the clavata3 promoter, a very specific part of the promoter, which is this distal promoter here, locule number per fruit instantly goes up. They did this by CRISPR-Cas gene editing. If they delete another part, so the proximal part of the promoter, nothing happens. So this distal part of the promoter seems to contain interesting functional elements for this locule number trait. So we in the lab tried to recapitulate this using Plant STARR-seq and deep learning. So we took the whole clavata3 promoter, chuck it again down into 170 base pair parts that we can use with Plant STARR-seq, and showed the strength of these individual parts, the enhancer strength of these individual parts, using Plant STARR-seq. And we can exactly see the same thing. The parts in this green distal part of the promoter here have a high enhancer strength. And the parts in the proximal promoter, they don't. Yeah? So these are actually the interesting functional sites that seem to have something to do with clavata3 expression. We tried to also throw our deep learning tools on it and to see if the predicted enhancer strength mirrors the measured enhancer strength, and it was near perfect. Like we could see the exact same pattern here. Very strong transcription or enhancers, very strong enhancers on the distal part of the promoter versus the proximal part of the promoter. And then now, as I said to you, we can open this black box here. We can now, by having this good deep learning model, we can now see, okay, what did the model learn? What does it think is important in these parts here? And we could find, again, like these patches of high importance for the model. And these were, again, transcription factor binding sites. And we think that these transcription factors do regulate the expression of clavata3 that leads to this interesting variation in the tomato phenotype. And with this, I wanted to show you how we can use these methods for interesting applications in agriculture and interesting traits. I promised you before that it should be easier to use these deep learning models also for your studies, because right now you would still have to have the model as a complex model file, and you have to use Python on a strong computer with graphics card access and so on to predict your own potential enhancer candidates. And right now I'm trying to make this easier by building a web tool that allows you to use our models, freely available models, to predict all certain kinds of regulatory elements. It's going to have three basic parts. The first is the predict part, so you can upload your sequences there and either get just a prediction, so how strong is the enhancer you put in there. But you can also use these attribution algorithms to open the black box again and to try to find interesting sites in your elements, in your enhancer elements, that could be transcription factor binding sites with high importance for the enhancer strength of your sequences. And if you do not want to plot this on your own, it's also going to feature a plot function that you can then use to plot your data and to make, hopefully at some point, publication-ready figures. Right now, this is in development, and we plan to publish this in, let's say, mid to late spring of this year. And then you should all be able to use this web server to analyze your gene regulatory elements and functionally annotate your gene regulatory elements. And with this, I'm done. I want to thank, first of all, the people from my old lab that I did my PhD in, the lab of Professor Andreas Weber. They are doing a lot of research on this interesting variation in photosynthesis. And then my current lab at the Institute of Synthetic Biology at HHU, doing Plant STARR-seq and very interesting deep learning stuff. I'm now again, and I was in my PhD phase, a member of the Cluster of Excellence on Plant Sciences at HHU Düsseldorf, which I have also to thank for funding. And with this, I'm done. I hope you could take a little bit with you, at least that Plant STARR-seq is very cool and deep learning is very cool. And I'm happy to answer some questions. So I start.